Hi, my name is Valerie Tran, and over the summer months of 2022, I was an intern at the Joint Genome Institute, or JGI, under the U.S. Department of Energy. The JGI supports genomic projects that help the Department of Energy mission areas related to clean energy. One specific area of interest is understanding the biology of a group of organisms called algae. I have been working with the JGI PhycoCosm page to do my research on algae and will be guiding you through a tutorial of the portal and how I used it to tackle my work. During this tutorial, I will go over an overview of PhycoCosm and how it can be used for research in the development of clean fuels. I will then go over a type of algae called Scenedesmus and an experiment to understand the regulation of genes in these algae. Lastly, we will walk through a case study to explore some individual proteins related to gene regulation and possibly the production of biofuels. First, we should understand what PhycoCosm is. PhycoCosm is an algal portal that was developed at the JGI to support and distribute algal genome sequences and other omics data, such as data produced from sequencing DNA and RNA. Here is a schematic of a phylogenetic tree of all the algal groups available. The tree also includes non-algal groups that are still relevant to the research of algae, such as Arabidopsis, which is a plant used as a model to guide other plant explorations. Now let's transition to the website. Here, we can click on a node on the tree to see the species under the group, and we can also see the other analysis features it provides. PhycoCosm currently hosts over 150 genomes sequenced. On the main page, you can use the search bar at the top if you know what species you are looking for, or you can scroll through the clade it's in, and the species are arranged in alphabetical order. One reason that the DOE and JGI are interested in studying algae is its potential as a source for biofuels. Diesel and gasoline are conventional fuels that produce harmful emissions when burned. They are a major source of harmful pollutants that lead to global warming. Changing to a renewable resource such as biofuels will lead to less fossil fuel burning. Biofuels are a fuel source that come from biomass, such as lipids. Algae creates lipids that can be turned into biofuels, and they are also photosynthetic, so they decrease the carbon dioxide in the air. Algae also use less resources than other land plants to grow, so that makes them a more sustainable option. Algae creates oils that can be turned into biofuels, but we need to find an efficient strain so that it can be mass-produced and someday cost similar to today's fossil fuels. While many species of algae are being explored for their use in biofuel production, in my research, I looked specifically at strains from the genus Scenedesmus. These strains here have the potential to accumulate oils, especially under environmental stress. We hope to understand promising strains of algae by looking at their genes and the transcription factors that act by turning genes on and off. Transcription factors are proteins that bind onto DNA at specific spots and regulates genes. Understanding these proteins can be useful for engineering strains at a larger scale commercial production. Scenedesmus belongs to the class Chlorophyceae. We can then click on the node for it and scroll down to look at a Scenedesmus of interest. After clicking on the name, we can see that it takes us to the home page where it gives us an overview of the algae. It was successfully cultivated in bioreactors and in outdoor ponds, which makes it a potential feedstock strain for biofuels. We want to look at what genes a transcription factor regulates. So we are going to look at DNA affinity purification sequencing data, also known as DAP-seq. This experiment allows researchers to identify where transcription factors bind in the genome by combining tagged transcription factor proteins with genomic DNA and sequencing where they bind. From the DAP-seq data, we can look for peaks in the experiment that shows the locations of where the sequencing reads are mapped. These peaks then represent the transcription factor binding sites. We can take these peaks and find out where they are in the genome. These location of peaks are then inserted into a program called MEME, where logos are then formed. Sequence logos are shown here, and this tells us where transcription factors bind and the sequence that these proteins recognize. This experiment was performed at the JGI using transcription factors from Scenedesmus. We can visualize the DAP-seq result on PhycoCosm. We can go to the Browse page here, where it will show us Genome Viewer display tracks. Each row is a different track which shows us where the nucleotide base position is along a scaffold. It shows custom tracks and more. Tracks here also show different data sets like DAP-seq data, shown from the green text on the left. We can see that the track in green represents DAP-seq mapping of a sequencing library. This corresponds to a DAP-seq experiment for one transcription factor. There, we can look for peaks. Each track corresponds to a different transcription factor, and they look very tightly packed at a first glance, but that can be adjusted based on the zoom bar at the top. The peaks on the track show the reads from the DAP-seq experiment aligned to the genome, which shows where the transcription factor is binding. Now let's go to a specific locus. Here, we can see one locus with clear peaks for us to look at. We see a spike in the DAP-seq data for this transcription factor, which correlates to a MIB-like transcription factor. A MIB-like transcription factor is a family of proteins found in plants and algae. It is one of the largest family of proteins and is involved in many pathways, including regulating responses to environmental stress. Clicking on this track here, and then clicking on Feature Web Page, will show us the sequence for the peaks above. This sequence can then be used to do further analysis in the meme suite. We now want to look at what genes are being regulated. Let's go down to the blue bars where we see the genes. This track shows the gene models. We can see exons, or the protein coding section of DNA or RNA, represented by the thicker blocks. On the left is a smaller block, which represents the 5' end, and on the other side is the 3' end. We can see the direction of the gene because we follow where the arrows are pointing. We can explore the gene further by clicking on it and then clicking on Feature Web Page. This brings us to the protein page, where we can look at the gene in greater detail, including information about what the gene does. We see that it is a diacylglycerol acyltransferase. This gene catalyzes the last step in triacylglycerol biosynthesis, which is the lipid precursor used for biofuels. Since the MIB-like transcription factor regulates this gene, maybe this transcription factor can be important for regulating synthesis of these lipids. Because it may be useful for biofuels, let's explore the family across more species to see if it is conserved. To do this, let's go to the drop-down menu under Annotations, and then clicking on Transcription Factors. There, we can look for our transcription factor of interest, the MIB-like transcription factor family, across other related organisms. If we want to keep exploring our transcription factor, we can scroll and look for it, or we can filter our results at the top. Each column represents a different species, and each row represents a different transcription factor. The number represents the number of annotated genes for that transcription factor family, and we can compare the number of MIB-like transcription factors across different organisms and get the list of genes in other species. We can choose one other species that we want to investigate further, and then click on the number under the species we're interested in. We can see the species by hovering over each number. After we click on the number, it'll take us to a list of genes we can choose. From there, we can see different features of the transcription factor, and clicking on the location will then bring you to another page for that gene to further explore other viewer display tracks and continue your research. Overall, we explored what genes a transcription factor regulates. We were able to efficiently find our species, then analyze data through the Browse tab. This then led us to clicking on a gene and understanding its function. We also further explored a transcription factor family. This research project continues to use PhycoCosm to see how transcription factor regulation is conserved across different species. I invite you to explore the function of other genes that are annotated on this resource or look at other transcription factors. Overall, PhycoCosm is a good resource for facilitating comparative analysis. This walkthrough was just one way to use PhycoCosm. I hope you explore the site further for your own research goals in the future. Thank you.